today speaks to this unique modeling approach that we have used here at Intellos. There are 3 different layers of physiology captured in the PhysioLab platforms. Here at Intellos, we use a unique top-down modeling approach aimed to capture the clinical outcomes, the top layer that can directly impact drug development decisions in the clinic. In the Metabolism PhysioLab platform specifically, our interest here, for example, is to predict changes of plasma glucose and insulin during fasting, after a meal, or following an acute therapy, as well as the long-term progression of A1C over time. These are the outcomes that, the outcome behaviors that our model should reproduce, which can be qualified against clinical data. The second layer is the physiological dynamics at tissue level, which are key biology whose interactions gives rise to the clinical outcomes. In the metabolism platform, we've captured major physiological systems necessary to represent the pathophysiology of type 2 diabetes. For example, the insulin-sensitive tissues such as the liver, muscle, and adipose tissue, the GI tract where we look at the digestions and absorptions of nutrients. We also look at gastric emptying and the secretion of gut peptides. We also look at the hormone secretion, including insulin, glucagon, epinephrine, cortisol, as well as the non-insulin sensitive tissues such as the kidney, brain, and other tissues. The third layer are key metabolic regulatory mechanisms that actually controls the adaptive networks of the tissue-level physiological dynamics. For example, including the hormonal actions of insulin and glucagon in insulin-sensitive tissues, biomarkers that either controls or respond to a particular pathway, etc. Those are the details necessary to reproduce rational tissue-level physiology in order to reproduce the clinical outputs that we're interested in, as well as the changes of biomarkers. While constrained by clinical outcome data, diversity in the complex interactions of physiological dynamics and the regulatory mechanisms allow the creation of alternative solutions in the underlying physiology, but still reproducing the appropriate outcome behavior. This is because the mathematical equations or the dose-response curves of hundreds to thousands of biological processes available in the metabolism platform are variable for different individuals due to genetic lifestyle differences contributing to differences in the underlying disease etiology. We can therefore create alternate virtual patients with alternate mathematical solutions. For example, 2 virtual patients with the same A1C level could have very different levels of the combinations of hepatic insulin resistance versus peripheral insulin resistance. And their combinations result in changes of fasting glucose and postprandial glucose that are different as well. Each unique solutions in the model generate 1 virtual patient, which I will be speaking about in further details later. But in a nutshell, multiple virtual patients can capture different patient phenotypes or similar or even identical phenotypes, but with very different underlying disease pathophysiology. And the platform and software technology we've developed in a way that allows us to create thousands of virtual patients very, very efficiently. What that allows us to do is to explore consequences and the clinical variability on decisions that you are making with regard to the patient target, particular combination strategies, optimization of the target, etc., before conducting a large clinical trial that is taking time and very costly. And this will help you save time, money, and mitigate potential risk going into the clinics. Keep in mind that the second and the third layers comprise a huge network of physiology where we have in a model around 800 state nodes capturing just the mass balances, 1,600 function nodes capturing regulatory rates and fluxes, as well as 22,000 parameters that we manipulate and utilize to compute the iterations of the mathematical solution, giving rise to the appropriate outcome behavior in virtual patients. The Intellos PhysioLab platform is a mathematical model of human whole body metabolism that enables the characterization of the pathophysiology of diabetes, as well as the prediction of the effect of diabetes treatment. And that's the major purpose or the goal of the functionality of the platform. The platform itself integrates quantitative data from thousands of scientific literatures or references into a single contextual framework of differential equations representing the dynamics of carbohydrate, lipid, and amino acid metabolism. Here, we have captured the biology of insulin-sensitive tissues, muscle liver adipose, the GI tract with nutrient absorption, gastric emptying, and gut hormones, hormone secretion, including insulin and glucagon, and insulin-insensitive tissues, including the brain, kidney, and other tissues. The current version of the Metabolism PhysioLab platform contains more than 3,200 references and published papers. A variety of in vitro, preclinical, and clinical data were used to inform the mechanistic structure, which means how the biological processes are actually connected. These data enable the modeling of physiological processes underlying the pathophysiology of diabetes, which I already touched on in the previous slide. The model tracks the levels of the biological elements. The simple example would be tracking plasma glucose and the rate of associated biological processes, for example, gluconeogenesis that is contributing to hepatic glucose output. Clinical data at the top level, at the whole body level, is primarily used to constrain and validate system-level behaviors of the model. For example, changes of A1C, glucose, insulin, or other measures that you could measure in a clinic. The physiological responses that we were able to capture and reproduce, including changes of a number of different markers during fasting or after a meal, or responses to exercise, manipulations or interventions including nutrient infusions such as OGTT, IVGTT, fat and amino acid loading tests, hormone infusions including insulin glucagon, and glucose clamps, energy and nutrient imbalance, etc. Virtual patients are a fundamental component of the Intellos disease models. Diverse patient types are available in the Metabolism platform that includes lean, overweight, obese individuals, healthy, pre-diabetic, and diabetic subjects, mild, moderate, severe disease phenotypes, and treatment-naive or patient treated with standard of care, including, for example, metformin, sulfonylureas, TZDs, etc. The Intellos Metabolism PhysioLab platform enables a direct head-to-head comparison for model and combination therapies with standard of care currently included in the model in the same virtual patient. This is not possible in clinical studies and extremely valuable for evaluating the efficacy of novel compounds under development. We were often asked of